Uh, Good morning, everyone, and thank you for joining us for today's webinar. I'm Jane Ackerman, and I'm the Programme Manager for the IUCN UK peatland programme. So I'll start today with some very brief housekeeping, and then a short introduction to the peatland programme and our biodiversity advocacy work before I hand over to the first of our brilliant lineup of speakers. Today's webinar is being recorded, but only our speakers and myself will appear in that recording. Can I just check with Liam that we've started recording?
Yeah, we're recording, Jane.
Fabulous, thank you. So we'll hold a Q&A session at the end of all of the talks. So please put your questions for any of the speakers in the Q&A feed. It'll also help me when I chair the Q&A session at the end if you let me know who your question is for. And it's fine if it's for more than one person, just put that in as well and we can share that. And you're also welcome to use the Zoom chat function during the webinar to make comments, introduce yourselves and where you're joining us from as well, but please don't use this to ask questions as they may be missed. So I'd like to start with a very brief introduction to the peatland programme for those of you who may be new to our work or perhaps aren't aware of our wider efforts to champion biodiversity this year. The IUCN UK peatland programme exists to promote peatland restoration in the UK and advocates the multiple benefits of peatlands through partnerships, strong science, sound policy, and effective practice. We were founded in 2009, and we're an umbrella organisation which represents a really broad range of partners, including government and devolved agencies, non-governmental organisations, researchers, and regional peatland partnerships, all working together for the restoration, protection, and sustainable management of UK peatlands. We provide specialist advice, and we advocate for land management and other policies which support peatlands and wider biodiversity. We also produce a wide range of publications and other resources to educate and inform people about peatlands and the issues which affect them. And we're continually developing the Peatland Code, which is a voluntary carbon finance standard for UK peatlands, which helps to fund restoration. Our vision is that peatland ecosystems are valued based on the many different ecosystem services they provide. Peatlands are our biggest terrestrial carbon store. They're home to unique biodiversity. They provide around 70% of our drinking water in the UK, and they help mitigate against flooding. They're also incredible archives of our cultural history, and they provide recreation, income, and joy for millions of people. Unfortunately, around 80% of UK peatlands are still in a degraded state, largely due to drainage for agriculture, forestry, and peat extraction. So there's a lot to do. But there are a growing number of dedicated peatland partnerships who are restoring and protecting these wonderful habitats by working together across multiple organisations, some of which you'll hear from today. Now, peatlands are receiving an increasing amount of attention for their role in sequestering carbon when they're healthy and emitting it when they're not. However, alongside the climate crisis, we're also facing a catastrophic loss of biodiversity. This year, the peatland programme has been raising awareness of the importance of a wide range of species through our species showcases, which you can find on the biodiversity pages of our website. We've looked at birds, butterflies, plants, and microorganisms so far, including their role in ecosystem function and what they can tell us about the health of our ecosystems and even their history. So if you have a peatland species you'd like to shout about, then please do get in touch. We also ran a webinar earlier this year on different approaches to biodiversity monitoring. And that's available on our YouTube channel, where you'll also be able to find the recording from today's webinar. So that's enough from me, but please do get in touch if you'd like to find out more about the Peatlands programme's work and how you can get involved. Now, I don't think it's possible to consider biodiversity and ecosystem recovery without considering species reintroductions. We saw in our large heath butterfly showcase earlier this year how impossible it can be for species with limited dispersal ability to return to areas of their former range following restoration work. And of course, for peatlands, the managed reintroduction of key peat-forming species is often essential to ensure that hydrological interventions actually result in ecosystem recovery. The International Union for Conservation of Nature, or IUCN, has produced some really useful guidance for those looking to use reintroductions as a tool in ecosystem recovery. And our first speaker today is going to introduce you to the principles within that guidance that can be applied across all ecosystems and not just peatlands. So Nida Al-Fulaij is CEO of the People's Trust for Endangered Species and also co-chair of the Species Survival Working Group under the UK National Committee of the IUCN. So Nida, thank you for joining us today. I'll stop sharing my screen now and hand over to you.
Thanks very much, Jane. I will attempt to share my screen. Is that full screen for everyone?
Yes, that is looking great.
Brilliant, thank you. Oh, well, thanks very much. So I don't need to introduce myself. I'll just briefly introduce People's Trust for Endangered Species. So we're a UK-based environmental NGO, but with global remit. So we fund a lot of conservation work and research on endangered species around the world. And then also here in the UK, some of those programmes which we lead. So we've been involved in Siamese crocodile translocations in Cambodia, northern river terrapin releases in the Sundarbans in India. And then closer to home, we've also funded the first official, part-funded the first official release of beavers at Knapdale in Scotland. We're currently supporting the Scottish wildcat release programme that's underway in the Cairngorms, and we supported the Vincent Wildlife Trust Pine Marten release in Wales. And we also lead on the hazel dormouse reintroductions here in the UK. So as Jane mentioned, you know that IUCN, it's the world's largest environmental network with over 1,400 members, mostly organisations, and over 16,000 experts. Established in 1948 and headquartered in Switzerland, it's an active network of scientists and experts that provide its members with conservation and policy advice. It's made up of 7 commissions which focus on different areas such as education, law, protected areas, but perhaps the most well-known is the Species Survival Commission, whose remit is to prevent the loss and aid the recovery of the diversity of life on Earth. The SSC is a science-based network of thousands of volunteer experts from almost every country in the world, and it's probably best known for the Red List. And the Red List is the most comprehensive list of the conservation status of animal, fungi, and plant species in the world. So the SSC gathers knowledge on species status, identifies the threats faced by different species, and then it develops policies and guidelines and advice to aid and drive conservation action. So they've produced many different guidelines, such as how to apply the Red List and guidelines for species conservation planning. But today we're focusing on the guidelines for reintroductions and other conservation translocations. So as Jane mentioned, I'll be talking to you about the principles of those guidelines, and then the other speakers will focus on putting those principles into practice. So what's the definition of conservation translocation? It's the deliberate movement of organisms from one site for release in another. And then these can either be reinforcements or reintroductions, which take place within a species' indigenous range, or they could be conservation introductions, and those comprise of assisted colonization and ecological replacement outside of an organism's indigenous range. But the overarching principle of a conservation translocation is that it must be intended to provide a benefit at the population or the species level, and it's not there purely to benefit the translocated individuals. And so, as this says on the slide, these guidelines are designed to be applicable to a full spectrum of conservation translocations. They're based on principle rather than example, and they're there to provide guidance on the justification and the design and implementation of any conservation translocation that people are putting into practice. So the guiding principles are that the translocations must yield quantifiable conservation benefits to the species or population that's the focus of the translocation, and they must be justified, which means there must be clear objectives. Anyone carrying them out must identify and make an assessment of any risks associated with the translocations. And there must be some measure of the performance of the translocation, whether positive or negative. And of course, with accelerated ecological change, specifically including changes to our climate, there are increasingly proposals to move organisms outside of their indigenous range. These are the assisted colonizations I mentioned before, and the number of these are expected to increase. Of course, they're more controversial and they're likely to pose greater potential risks than traditional reintroductions within a range. But of course, all conservation translocations come with their own risks, and therefore the guidelines recommend that all alternatives are considered before a translocation does take place. So just to go over the definitions, which are a guiding part of the principles, we've got translocation, the overarching term, which is human-mediated movement of living organisms from one area for release in another. So these can be living animals moved from one wild location to another. They can be captive bred animals or other organisms that are moved into the wild. And translocations can, of course, be accidental, such as stowaways, or intentional for a variety of different reasons, including welfare or commercial. So then when we frame it under the term conservation translocation, this narrows the definition, and it's the intentional movement and release of a living organism where the primary objective is conservation benefit. So these will usually comprise improving the conservation status of a focal species either at a local or a global level, and or restoring natural ecosystem functions or processes. As I mentioned, they can happen within or outside an indigenous range. So the indigenous range of a species is defined within these guidelines as the known or inferred distribution generated from historical, either written or verbal records, or physical evidence of its current occurrence. So we've got two different types of conservation translocation. There's population restoration, which I'll talk about first, and this is when the translocation happens within an indigenous range. So first off is reinforcement, and that's the intentional movement or release of an organism into an existing population. And these can be carried out to enhance a population's viability by either increasing its actual population size, increasing its genetic diversity within a population, or even increasing, for example, a specific demographic group or stage, such as either males or females, or juveniles or adults within a population. And then there's the reintroduction, which is the intentional release of an organism inside its indigenous range from which it has disappeared. And the aim here is to establish a viable population of the species in question within its indigenous range. And just as a brief example, PTES has done both reintroductions and reinforcements of hazel dormice as part of the species recovery programme. So predominantly we captive breed dormice with some rescued animals from the wild and release them. So since 1993, we've carried out releases at 25 different woodlands in 13 different counties, so restoring the population up to the Scottish borders where they were once found but had been lost in the last century. And then we've also revisited some of those early releases and carried out some reinforcements to ensure there's a bit of genetic diversity, because often we re-establish a population in a woodland that's relatively isolated. And then we go on to the second kind of conservation translocation, which is a conservation introduction, which is the intentional release of an organism outside its indigenous range. And these can be assisted colonisation, which is the intentional movement and release outside its range to avoid extinction of the populations of focal species. And this is normally where protection from current threats in the current range is likely to be less feasible than at alternative sites. And it can either involve, a small incremental range extension or an actual move of these organisms quite far from their current range. And then the second type of conservation introduction is ecological replacement. And that's when there's an intentional release of an organism outside its indigenous range to perform a specific ecological function, as Jane touched on earlier. And these can be used to re-establish functions. And at that point, we might identify the most suitable existing subspecies or a close relative. So, for example, here's a picture of an exotic giant tortoise in Mauritius, which has been introduced to an island to restore the ecosystem functions because they're considered keystone grazers, but the original population of giant tortoises became extinct. So when is a translocation acceptable? We need strong evidence that the previous threats have been identified and removed. We need to consider the benefits and the negative impacts and ensure these have been identified and considered. We need to consider the absolute level of risk, and that needs to be balanced against any potential scale of expected benefits that's brought about. Where there's a high degree of uncertainty or it isn't possible to assess that a conservation translocation presents low risks, then the guidelines advise that it shouldn't proceed and alternative conservation solutions should be sought. So just to kind of bear in mind, obviously, beaver reintroductions are very popular and at the forefront of discussions in the UK at the moment. The arguments on one side are that the positive benefits will help reduce flooding risk in many areas of the country. But on the other hand, we've got to weigh up what the negative impacts might be at a local level with damage to private landowners. So conservation planning, this schematic's within the guidelines, so accessible to everyone. And the Species Survival Commission approach in conservation planning requires that goals are set, objectives are defined, and actions are detailed. So there should be an overarching goal, which is a statement of the result, and it's often expressed in terms of a desired size or number of populations that will achieve the conservation outcome, either at a local or a global scale, and all within an overall time frame. And then the objectives will detail how the goal will be realised, and they'll be clear and specific with details of actions that need to be taken to meet the objectives. So, for example, one thing we do, another conservation translocation programme we've been heavily involved in is water vole restoration, but of course the key objectives would be to ensure that any site is mink free before water voles are released. Other parts of the guidelines advise that monitoring and exit strategies are well detailed, so each stage of the translocation should be monitored. It should be clearly outlined what evidence will be measured on progress, what the data, what type of data needs collecting, and where and when to provide that evidence, and information about who's going to collect and analyse the data, and then who's going to disseminate the findings, so that learnings, whether things are successful or not successful, can be widely shared. And of course, exit strategies are important as well. You know, we need to consider whether investing further resources is no longer justifiable if either a lack of success or undesired consequences are coming about as part of a conservation translocation programme. The next thing we advise is to look at biological feasibility. One of the first requirements is, of course, habitat. It might be that previous range might be unsuitable, or the best habitat to return a species to isn't available locally. So the habitat needs at a site must be considered carefully. They must match the availability of the needs of the candidate species, and so that's central to the feasibility and design of the programme. Climate should be factored in, and obviously that's ever more crucial at this stage and ever more difficult to quantify in some instances. So the climate should be suitable for the foreseeable future, which can of course pose challenges in identifying suitable sites. Another key consideration is about the founder population, which, as I mentioned previously, can be wild or captive bred. The founder's genetic source should be, the genetic diversity and the source population are also important to consider. So it's recommended that the founders should show characteristics which are based on the genetic provenance and morphology, etc., as close as is appropriate with the original population. Any potential negative effects of removing individuals from a population, either in the wild or in captivity, should also be assessed. So, for example, we supported work on the source population of pine martens in Scotland that provided the individuals for release in Wales, to ensure that the removal of individuals over a series of removals didn't actually negatively impact the genetic diversity at the founder site. And of course, source populations ideally should be physically closer to or from habitats that are similar to the destination site that's in question, so that they're more genetically suited. But it's also important to note that if founders from widely separate populations or areas are mixed, there could be genetic incompatibilities, and that's something to consider, because there obviously may be very few individuals or few populations left, which is part of the reason why conservation translocations are being carried out. So these are all things that need to be weighed up and balanced. We must also consider the welfare and disease risk to the organisms that are being translocated, which can be quite diverse and quite contradictory. So it's important that the group carrying out the translocation attempts to adhere to internationally accepted welfare standards, and also comply with any legislation and policies that are in place, both in the source and the release area. It's critical to make every effort to reduce the stress and suffering during the whole process. So that can involve capture of wild animals and handling them, transporting either wild animals or captive bred animals to the site and the holding facilities. And stress can come about, of course, through confining unfamiliar individuals in close proximity, both up to and after the release. Many of you will be aware of the phrase soft release. So we do that with the hazel dormice. When we take them on site, we put them in cages, and we ensure they're fed and get used to their surroundings before we open a small door in their cage to let them out into the wider woodlands. So the soft release is an idea to give them, to let them get over the stress of the transport and settle in. But of course, these soft releases can, of course, increase stress if you're moving wild caught animals and prolonging their captivity before a release. And source population individuals can also suffer stress due to the removal of individuals disrupting established social relationships. And as well as welfare, of course, a critical thing to consider is disease. But the principles do recommend that disease risk should be proportional. Quarantine is, of course, a basic precaution that's often used in conservation translocations, but it can sometimes cause unacceptable levels of stress. So these have to be balanced out. The next crucial thing to consider, of course, is the social feasibility of a conservation translocation, not just the biological feasibility. So the human communities around the sites where the individuals are going to be translocated are going to need to be engaged and listened to, and their concerns considered. Obviously, translocating wolves to the UK is something that's been discussed for some time, but fills many people with alarm, and so of course community attitudes can be very extreme and contradictory. So the translocation planning process should really accommodate social economic circumstances, the attitudes of the local community and their values, any motivations and expectations, and any anticipated costs and benefits to the community in significant detail before any translocations go ahead. It might be that local communities have no connection to a species that's being proposed for release, and hence they might be in opposition to a release being carried out. But of course, conversely, successful translocations may yield economic opportunities, such as through ecotourism. But as I mentioned previously with the beavers, there can also be local impacts to individuals or to wider groups that need to be taken into account. And just finally, there's a few other considerations to think about. Regulatory compliance, so all levels of legislation need to be taken into account. For example, when we release hazel dormice, if the site in question is a triple SI, we'll need to get special permission to do it because the site might have been designated for another species. And of course, international movement of organisms can be particularly complicated. I'm not sure how many of you know about the quite high-profile translocation of cheetahs to India. Another thing to consider that was a big factor in that release was, of course, the resource availability and the funding. And it sounds obvious, but conservation translocation shouldn't go ahead without the assurance of all funding in place to cover the release and any exit strategies that are needed. And of course, risk assessments are critical. They're detailed quite broadly in the guidelines, but I won't go through them. But they are obviously context specific and need to be given full consideration. And then, of course, if you've taken all that into account and still decided that a translocation is right to go ahead, you move into the release and implementation phase. And of course, so there are other very key considerations to take into account, which is the release site and the habitat that's there and whether it's going to meet the requirements of the organisms, but also whether there's adequate connectivity so that those organisms can move into the broader countryside and expand the original population. The release strategy, how many animals are you going to release? What will be the sex ratio, or in terms of plants, how many founder individuals are needed? And how many sites are you going to release them at? Is one sufficient, or should there be a cluster? Do they need to be widespread or in close proximity? And monitoring, of course, is crucial. So you might detail out different stages of success of a programme based on whether the organisms reproduce at the site, whether those offspring also reproduce in later years, whether those individuals move beyond the original release site or increase the number of individuals in the population. And it's also critical to continue to look out for new threats and monitor the health and the behaviour and the genetics of the population in question. So because of this monitoring, obviously, it's recommended that it's a monitoring and management approach that's taken to the site. And of course, finally, it's important to disseminate results and share learnings widely, so that we can learn what works and what doesn't work. So on that note, I will say thank you very much. And I think we're going to wait for questions until the end.
Thank you, Nida. That was a absolutely brilliant introduction to those principles. And a fantastic start for us to move on and start looking at some of the examples of these reintroductions that have been happening in the peatland space. So I will hand over now to Tom Spencer. Tom is the Senior Research and Monitoring Officer at Moors for the Future Partnership, and he is going to do exactly that, disseminate some findings, the last bit on Nida's slide just there. And he'll be talking about Moors for the Future's work to reintroduce sphagnum to the uplands of the Peak District. So I will hand over to you, Tom.
Great. Can everyone see my presentation?
Yeah, that's looking great.
Yeah. Great. Okay, excellent. So yeah, thank you very much. So I'm Tom Spencer from Moors for the Future Partnership. And I'm going to be talking about species reintroduction with sphagnum moss as an example. So yeah, I'm talking about the Peak District National Park and South Pennines. I'm just going to give you like a very brief overview of the presentation to give you all the spoilers right at the beginning, so there's no suspense. So I'm going to cover 4 questions overall, all about sphagnum reintroduction. So firstly, sphagnum reintroduction, is it needed? The answer is yes. Does it work? Yes. Does it do good things? Yes. And do we need further research? Yes. Okay, so you can all just go and get a cup of tea now if you want. But yeah, so we do conservation work at a landscape scale in Moors for the Future. So we're working across a large area, you can see on the map here. And I think a key thing when thinking about why we need to do conservation work in the peatlands in this area is about the human populations around them. So you can see on this map that our work area is surrounded by large habitations, which crucially during the Industrial Revolution were pumping atmospheric pollution up into the sky from all sides. So this generated large amounts of acid rain, which fell onto the peatlands, whichever way the wind was blowing, and it rains a lot in this area, as anyone who lives around here will know, generated a lot of pollution, so heavy metal contamination and acid rain, which killed off large swathes of the sensitive vegetation in this landscape, so including sphagnum, crucially. So why are we intervening in this landscape? Because it's an environmental catastrophe, essentially. So we had high air pollution, wildfires which were not put out because there weren't the resources to deal with them, so they were just left to burn. Severe weather events exacerbate the damage already caused by the air pollution and the wildfires. So we've got, in particular from the acid rain and from the wildfires, we've got large areas of completely denuded landscape, so all the vegetation killed off, and because of the high levels of contamination, plants were not regenerating themselves, so large areas remained bare, and then on top of that you get weather events which can then erode the peat surface, which when it doesn't have any vegetation on is pretty fragile. So then you start getting erosion of the peat surface, which then forms a highly dense drainage network, much more dense than it would naturally be. So you get deep erosion gullies and lots of them. So large loss of peat. Peat is roughly 50% carbon. So we're talking about large scale loss of carbon with obvious significant impacts and implications for climate change. So the story of how we've got to the degradation of the peatlands in this area has various nuance, but I think they're the most key things. And they led to a situation where we had massive areas of completely bare peat. So like in this image, about 35 square kilometers of completely bare peat in our work area. And bare peat which was not rehabilitating itself and was just continuing to erode. So we're talking around 2000, 2003, around then. That's around the time that North of the Future Partnership started. And since then, we have done 35 square kilometres of bare peat restoration. There is still a bit more to do, but we've tackled the big chunk of that. And we've worked over a much wider work area, so 250 square kilometres of blanket bog restoration. So not all of it was bare peat. So some of it has slightly less severe degradation. It still has vegetation, but still needs work doing to it. So it's a massive operation that North of the Future has been going through. And depending on the needs of the site, we do different interventions. So the key thing to start with is stabilizing any unstable areas. So if that's areas of bare peat, then getting something to grow on them just to stop the erosion. And re-wetting is key. So we do lots of gully blocking using different techniques depending on the site needs. And then once we've stabilized the habitat and improved conditions hydrologically enough for sphagnum to be sustained, we will then plant sphagnum. We'll talk more about sphagnum in a second, but just continuing a quick overview of what Moors for the Future is and does. We have a science team who, myself included, monitor the impacts of the restoration work that we're doing and conduct sort of research trials to work out what the best ways are to do our conservation work and inform changes to best practice. And then we talk about it as much as we can to anyone who will listen, because it's a very under-knowledged area socially. And obviously, if we find out important findings through our research, we want to tell other people about that, so that as a whole community, we can move forwards with peatland restoration in the most effective ways. Okay, so moving on to sphagnum. Sphagnum is important for peatlands because it's a keystone blanket bog species. It can hold up to 20 times its dry weight in water, and that means that it holds water on the peat surface and protects it from the erosion that we've just been talking about. Because it holds water on the surface, it creates anaerobic decomposition conditions, which means that it is able to build peat. That's how peat is formed. It's through anaerobic decomposition. So we need a saturated surface for peat to form. Sphagnum is a good way of achieving that. Obviously, surface pools can do that as well, but sphagnum is important where we're not in pools, and it will also grow in pools as well. It needs wet surface conditions, so you can't plant it directly into dry bare peat, which is why we have to restore the habitat to some extent hydrologically first before we can reintroduce sphagnum. So you have to get the conditions right first. And crucially, there was hardly any at all in the Peak District and South Pennines before our restoration work. Going back to that slide of the large expanse of bare peat, there's obviously no sphagnum there, and sphagnum is not going to grow in those conditions. So we'd lost almost all of the sphagnum that should have been there in the Peak District. So it was necessary to bring sphagnum back in, but we could only do that once we got the habitat and the landscape to a certain condition. Okay, so again, if you kind of imagine that that first bare peat image that we looked at, lots of people were saying at that point, sure, we need sphagnum here, but surely it's never going to work. Like the form of this landscape is so heavily degraded that surely it's not possible to bring sphagnum in. And More to the Future and various other organisations were like, well, let's give it a go. What have we got to lose? So on Kinder Scout, we started our first trials into sphagnum reintroduction in 2015. And so that was 5 years after we'd started revegetation work up there. So we started by planting amenity grasses, essentially, and then allowing a natural succession process to follow from that. Accelerated a bit by planting in some natural moorland species, but largely letting succession take its course. 4 years down the line, 5 years down the line, we're then like, okay, let's try putting some sphagnum in, just see what happens. We and everyone else were pretty amazed by the success of that, given that we weren't sure if it would work at all. We found that where we've monitored it, we know where exactly where various plugs were planted them into quadrats, and we've gone back in subsequent years to see if they're still there. We found a 90% survival rate of planted plugs. That is specifically into sites that were bare peat and have been revegetated, so heavily degraded sites. But with a new succession process taking place. So sphagnum has got in there and is able to tolerate the conditions, and 90% of plugs have survived. Not only have they survived, they have thrived. So I've got two lines on this graph. The lower one, I don't know if you can see my cursor, but the lower one with the circles on it, that is sphagnum monitored on undulating ground, so in quadrats on undulating ground, so not necessarily the wettest bits of the site. This is on Kinder Scout again. Not necessarily in the wettest bits, but including some wet areas. And in 9 years after planting, is that 9 years? Maybe it's slightly less than actually, yeah, it's 7 or 8 years. We've gone from 0% cover up to about 35% cover, which is pretty remarkable. We planted at high density, but even so, you can see the growth rates are pretty astonishing there. And the steeper line with the triangles, that's where we've done surveys in the wettest areas. So in the flow pathways where you would expect sphagnum to do the best, we're now up to about 90–95% cover in those flow pathways. So really outstanding results, which have blown all of us away. So this is all about sphagnum reintroduction into previously bare peat sites. As I said, a large proportion of our work has moved on from that bare peat revegetation onto diversification of sites which do have vegetation, but sites which are dominated by single species of vegetation. So we looked at 3 different types of vegetation: dense heather. Oh, sorry, I've jumped on a slide. I've jumped ahead of myself. So I'm going to go back to Kinder. So as part of our planting on Kinder, we wanted to try out a few different ways of reintroducing sphagnum. So one is from hummocks. So that's finding where we could find naturally existing sphagnum in large enough areas to translocate some of that. We've taken small samples of that sphagnum out, transported it up onto Kinder, and planted it into the ground, into these quadrats. So that's the hummocks. And we planted plugs, so they're grown in a nursery in a greenhouse. So small, they're about the size of a 50p coin. Plugs of sphagnum with 11 different species of sphagnum in them. And then slime, so that's just spores and fragments of sphagnum put into a gel, which you can then apply direct to the peat surface. So we're trying to work out what's the most effective and what's the most cost-effective way of reintroducing sphagnum. And you can see from the graph that all 3 have actually done fairly well. The plugs are performing the best. So we've gone from pretty much no sphagnum cover in the quadrats at the time of planting up to large areas of sphagnum grown from these tiny little plugs. The slime has actually done better than we thought it might do, and has caught up with the hummocks. The hummocks seem to be slower growing. We're not sure why, but the plugs seem to be the best, and we thought that they would be the most cost-effective method, and they do seem to be. Okay, so now we are moving on to the sites dominated by different species. So we looked at sites dominated by dense heather cover, by dense cotton grass cover, and by dense purple moorgrass cover. So they're all sites where there was hardly any sphagnum on the site at the start, and we wanted to see what would happen if we planted sphagnum into these sites. I think the most interesting one here is the heather, where after a relatively slow start, the sphagnum has really taken off in the last few years and is now doing better than on the cotton grass site, which is a little bit of a surprise, if anything, because heather creates quite a dense canopy, and it's generally assumed that sphagnum won't grow very well under dense heather, partly because of shading out and partly because heather creates slightly drier soil conditions than other plants. So the fact that the sphagnum is doing really well in dense heather is a surprise and is good news in terms of the possibility of diversifying the large areas of heather-dominated moorland that we have at the moment. So that's a really encouraging finding from the last few years. Okay, so sphagnum reintroduction, is it possible? Yes, in a whole range of different conditions that are potentially quite hostile to sphagnum, you might think. So that's really good news. The next thing that I want to talk about is sphagnum resilience. So as I mentioned before, it's a fragile and sensitive species that needs particular conditions, largely around availability of moisture, and was almost entirely wiped out by industrial pollution. So we, as bad luck would have it, set up a trial at Marsden Moor to look at planting different types of sphagnum. And I think 6 weeks after we planted the sphagnum, a severe wildfire burnt through Marsden Moor and wiped out pretty much everything that was there. The site looked like this in May 2019. And you can see that the bryophyte layer, so the mosses of different types, were almost completely eradicated along with all the other vegetation. You can see some of the cotton grasses starting to grow back there already, but the bryophyte layer has largely gone. What we found on that same day was that there were patches of sphagnum which had survived. So it looks pretty fried, but inside there are little green bits. So there was, I think because the sphagnum holds a lot of moisture, it had weathered the fire a little bit better than some of the other bryophytes. So that was quite interesting to start with. Here's a map of the wildfire burn scar. So you can see these 4 plots of quadrats that we set up with the different sphagnum mixes. And so the fire completely burnt through 2 of our plots, burnt through half of one of the plots, and didn't touch the site where we had our control. So we didn't plant any sphagnum in those quadrats. But this then gave us an opportunity to look at whether sphagnum can survive a wildfire. And surprisingly, it can. So we monitored the quadrats a couple of months after the wildfire, and we found 236 out of the 240 plugs that we'd planted were still there. We weren't sure how long they were going to continue being there, but they were there at that point and even had put on some growth. And then we went back this year. I processed these data 2 days ago, so they're quite hot off the press. And we found that, so the controls at the bottom, there was no sphagnum there at the start. There's still no sphagnum there. These 2 here, the pale blue and the purple dots, are from the quadrats which were completely burnt, and the green dot is from the quadrats where about half of the quadrats were burnt. All 3 of them have put on significant growth. The green one is doing the best, so there's some indication that the fire has inhibited recovery of the sphagnum, but the sphagnum has recovered at all the quadrats. So that's really encouraging. We weren't expecting it. So sphagnum is more resilient than you might think, which suggests that the damage historically was potentially more from industrial pollution than from wildfire, potentially. Okay, so that's so we've covered sphagnum reintroduction. Is it needed and does it work? Yes, and it is actually quite resilient. So I'd like to move on now to some of the impacts of that sphagnum reintroduction. So this is, does it do good things? And again, the answer is yes. So we monitor hydrology, we monitor water table, soil moisture, and stream flow. And I'm going to focus most on stream flow here because this is where the biggest hitting benefits are. So we measure stream flow at our experimental sites on Kinder, where we've done the revegetation of bare peat and then dense sphagnum planting. And we measure stream flow through V-notch weirs like this. And what we saw before we did any restoration, so this is when everything was bare peat, on the left-hand side of this graph, we get very flashy hydrographs. So you get a bunch of rainfalls, the level in the stream goes up very rapidly, almost in sync with the rainfall. The peak of the storm in terms of rainfall synchronizes with the peak of the discharge from the catchment outlet. And then as the storm dies down, the stream level drops back down as well. So before restoration, all three of our monitored catchments were behaving the same. So this is one storm from our baseline period. So the black line is where is our control site, and the green and blue lines are our restoration sites. But at this point, there had been no restoration done. So that showed us that all three catchments were behaving in the same way when they were bare peat. We then revegetated, we did some gully blocking, and at the blue site, we then did sphagnum planting 4 years later. And so then on the right-hand side of the graph, this was 2021, so 6 years after the sphagnum planting and 10, 11 years after the revegetation work, you can see that the black line, the bare peat control, where we did no restoration, the peak of the storm is still pretty much in sync with the peak of the rain event. But the blue line, which is where we did revegetation, gully blocking, and sphagnum planting, has a completely different hydrograph response to the storm. So the peak of the peak flow is much lower than at the bare peat control site, and it occurs much later than at the bare peat control site. Both of those things are important for reasons we'll come on to. So just to summarize those findings, in the baseline period, so I'm just talking about this blue line site where we did revegetation, gully blocking, and sphagnum planting. In the baseline, the lag time, which is the time from peak rainfall to peak discharge, was 30 minutes. Once we put in the gully blocks, that increased to, that increased to 80 minutes. And once we'd planted the sphagnum and that had increased to about 35% cover, that increased to 200 minutes. So 30-minute lag time in the baseline, pretty much in sync. We've added 50 minutes to that lag time by putting in gully blocks, and then that's gone up to 200 minutes once we've got dense sphagnum cover. So it's really significant change. And at the control site, we're still around 30 minutes lag time. So it's big change caused by that sphagnum. And that is because the sphagnum, as I said before, can hold up to 20 times its weight in water, can grow up to, well, at the moment it's approaching 20-30 centimeters depth on top of the peat surface. So it's creating this really rough, wet surface on top of the peat. And so when rainwater falls on that, it has to move through all of that wet, dense sphagnum, and it's going to take a long time to get into the streams. And that means that its travel time from landing on the ground to getting into the streams and rivers is significantly extended. It takes longer to get off the hill. So that's interesting at a headwater catchment scale on Kinder. But what we're really interested in is what happens at places that might get flooded. So, for example, in our work area, Glossop is a town underneath Kinder Scout, which has flooded quite severely in the past. So we're interested to know whether our restoration efforts on Kinder are going to impact somewhere like Glossop, where like flooding can have quite significant economic implications. So we're not able to do our dense sphagnum planting like we've done at that mini headwater catchment on Kinder across all of the land above Glossop, because there's a lot of land to go at and there are very complex landowner issues. So modelling is the way that we've gone about this. So the University of Manchester led a project called Protect, which ran a modelling exercise with a huge level of complexity. And they took the data from Kinder and they extrapolated that up to model what would happen in Glossop if we were to do the same intensity of restoration efforts across all of the available land in the Glossop catchment. So they came up with a scenario of revegetating all of the available bare peat and doing all of the gully blocking that would be feasible, and planting all of the suitable land with sphagnum. And then they worked out what would likely happen down in Glossop, and the results are really quite astonishing. So they were looking at flood relevant events. So that's they categorized that as 10 to 1,000 year return period storms. So a 10-year return period storm is a storm you'd expect to happen once every 10 years. And a 1,000-year return period storm is a storm you'd expect to happen one in 1,000 years period. So they're really, really big storms, the kind of things that will flood somewhere like Glossop in current conditions. And they found that if we were to do all of the restoration works, then we would expect that peak discharge in Glossop would be reduced by 23 to 51% compared to what happens at the moment. So potentially reducing flooding by a half. So that's peak discharge. The highest that the river in Glossop would get would be reduced by half in flood relevant events, which is I can't explain how massive that is. You know, the margins when we're talking about flooding and damage are pretty small. You know, it's if the river just stays within the concrete flood defences, there's no damage. If it just gets over those flood walls, we get a lot of damage. So reducing the peak flow by a half is absolutely massive, and we would get a 90 to a 98% likelihood of reducing peak discharge by more than 5%. So it's almost definitely going to reduce peak discharge by at least 5%, which even that is massive. And on average, we would expect peak discharge to be reduced by 23 to 51%. Obviously, they've looked at a massive range of different kinds of storms, so that's why there's a range in those percentages. And the big reason why we would see those reductions is because of that increase in lag time. So essentially what happens if you attenuate storm flow up on the tops is that you desynchronize the delivery of floodwater into Glossop. So we've obviously got a large area of lowland around Glossop, and then we've also got the upland peat. If you can slow down the water from the upland peat, it's going to take longer to get down, and it's going to come through Glossop in a separate flood wave to the rainwater from the lowlands. Without restoration, the water from the uplands comes off so quickly that it catches up with the lowland flows, and they all go through Glossop at the same time, which is why we get a massive flood wave. But by desynchronizing it, by slowing down the flow in the uplands, then we get two separate flood waves, potentially 3 or 4, depending on travel times from different parts of the catchment. And that means that no one flood wave is anywhere near as big. So these findings, like I said, they're absolutely massive. In terms of sphagnum specifically, they pulled apart where those impacts were coming from, and they found that if you just do revegetation and gully blocking, then you would reduce peak discharge in Glossop by about 5 to 16%, and by adding the sphagnum, you increase that to the 23 to 51%. So the sphagnum is having a really massive part to play in that flood protection, essentially. So like I say, this is all very new. The paper that this has been published in has only just come out in the last month or so. So it's very much stuff that we're finding out at the moment. Okay, so that's covered. Does it do good things? The answer is yes. So there are lots of other benefits, you know, to biodiversity, protecting the peat from erosion, improvements to water quality, that kind of thing. But I thought that talking about this flooding impact, I think it's the most sort of big hitting thing, arguably. So sphagnum, do we need further research? Yes, there's a lot of sphagnum happening. You know, this work season, so this winter, Moors of the Future is organising the planting of about 2 million plugs. So it's a thing that is already going at pace. But there are lots of questions that no one really knows the answers to, like how should you plant it? At the moment, we're planting individual plugs in places that we think it will do well. But there's a few options for different species mixes. How big should the plugs be? Does it matter? Will they be more resilient if the plugs are bigger? We plant at the moment in wet conditions, but is it possible to plant in dry conditions? You know, we've seen from the Cluna site that we can plant into dense heather, so can we plant into dry areas? Does the peat need to be 40 centimetres deep for us to plant into it? That is an assumption at the moment, but maybe if you get thin peat which is wet, we could plant into that as well, maybe. So we're starting trials at the moment, and obviously continuing existing research to look at these questions, and also replicating this experimental work on Kinder at a new project just close to the A57, where we're also looking at stream flow, water table, and soil moisture. And I mentioned biodiversity. We've started monitoring impacts of restoration and sphagnum on things like moths and aquatic invertebrates and bats, and we're finding some quite encouraging results, but that's all research in its infancy. I think I might have gone over in terms of time. Very sorry about that. And I have now finished. So yeah, just in summary, those 4 questions. Sphagnum reintroduction, is it needed? Yes. Does it work? Yes, even in difficult conditions. Does it do good things? Yes. And do we need further research? Yes. Thank you.
Thank you, Tom. We'll let you get away with going over because that was such an interesting talk, and it's wonderful. I made it up Kinder Scout for the first time myself earlier this year, and it's incredible to see what you guys have achieved up there. And it's also so important to have all of that monitoring behind it and the evidence to show what's happening and the impact of what you're doing. So brilliant talk, thank you. So moving swiftly on, I'd like to introduce Stuart Green, our next speaker. Stuart is the entomologist for Citizen Zoo, and he's going to talk about their work to reintroduce the large marsh grasshopper to the wetlands of East Anglia with some very helpful community keepers as well. So over to you, Stuart.
Well, thanks very much, Jane. I'm also a technological dinosaur, so just bear with me. I think this is going to do it. No, I don't want that. Okay. Okay, and I hope you can hear me. So this talk is about a collaborative project led by Citizen Zoo, which has been running now amazingly for just over 6 years. When we started, it was exactly 50 years since this species had last been seen in East Anglia. The large marsh grasshopper, or LMG, as we call it, is our largest and most handsome species of grasshopper in the UK. This is the common, but it's all, unfortunately, it's also one of the rarest. This is the common colour form you can see here, but it also comes in a rather fetching shade of pinkish purple, which is only found in the females, and it's a beautiful grasshopper. The species is restricted to very wet habitats where much of the substrate remains saturated all year round. It has an annual life cycle. The eggs, which are laid in pods in the summer, they're laid into the soil in pods of 10 to 15 eggs inside a hard foam coating, into the soil surface or amongst the bases of tussocks or other low-down vegetation, including sphagnum. The eggs are uniquely sensitive to desiccation, and I think that is the key thing with this species that really limits it to peaty habitats. This is what we'd consider to be a typical large marsh grasshopper habitat in the UK. It's a valley mire or lowland bog. It's acidic, nutrient poor, and has an abundance of sphagnum and other bogland plant species. However, in the rest of Europe, and it also occurs in quite a few other habitats, including fens, riverine pastures, and wet heathland. And it used to be the same in the UK. If we look at this map here, the dark spots show the current distribution, which is nowadays centred around in and around the New Forest and a little bit to the west of that, with a small resurgent population in the Somerset levels. But until recently, large marsh grasshoppers hadn't been seen in East Anglia for 50 years. And the white spots show sites where it was previously found, and we know these from museum specimens and from in the natural history literature. Most of the sites where the large marsh grasshopper has been lost are former fenland peat sites, and this project believes that they have the potential. These sites have the potential to support large marsh grasshopper again. Now, this slide just is a sort of overview of the habitats in East Anglia where it used to occur, and there's three separate areas really. West Norfolk, there's a series of peat bogs to the north of King's Lynn running up there. So that's very much the sort of typical New Forest type habitat. But there's also two other areas, the Norfolk Broads, where the last large marsh grasshopper was seen in 1939, and the Cambridgeshire Fens, which obviously were drained a lot earlier. And and both the Broads and the Cambridgeshire Fens are fenland with peat-rich soils. The area of the Cambridgeshire Fens where the large marsh grasshopper used to occur are what we call black fen areas with a high soil peat content. There are no records from further to the west and the east where it's more of a silty, clayey substrate. Now, the idea of reintroducing the large marsh grasshopper into East Anglia is not a new idea. I did my PhD in grasshopper ecology at UEA 40 years ago, and I was thinking about it then. It wasn't what I was doing my research on, but I did wonder, why did it disappear and could it come back? And at that stage, it probably couldn't, because a lot of the habitats were simply not suitable anymore. And what has made the current project feasible now has been a program of habitat restoration at two national nature reserves in West Norfolk, which has been conducted by Natural England and the Norfolk Wildlife Trust. And basically what they've done, they've taken many of the measures that we're talking about just now to reduce the drainage and raise the water table. And what this has resulted in is an increased growth of sphagnum and a resurgence of many of the other typical bogland species as well, and an increase in the area of these plants. So the actual area that could be suitable for large marsh grasshoppers has greatly increased at the two sites. So very good. Now, our project was conceived in 2017-2018, and the aim is to re-establish large marsh grasshopper populations away from the New Forest so that we can give the species better resilience against climate change in the future. And we considered two approaches. The first one, in principle, the simplest, was to do a direct translocation. And the kind of drawback with that is that it is a near endangered species, so we couldn't take too many individuals from the New Forest to translocate them. And there are always a lot of chance factors that can affect the success of an individual translocation, and quite a lot of unknown unknowns, and also unknown unknowns, if you like. What we also considered was whether it be possible to have a captive breeding program and actually breed wild-caught grasshoppers in the laboratory or in locust cages under continuous electrical sunshine with a superabundance of nice fresh food and no predation. And by that means, get them reproducing at a really good rate, producing lots of egg pods, which can be collected and processed, then used to produce grasshoppers the following year that could be introduced. So if we could do it, that I think was our preferred way. But we didn't know at the start. There's rather limited information on this species. I've worked a lot in the past on rearing grasshoppers and locusts, but this was a new one to me. And it's a species that lives in a very wet, boggy place. So we didn't really know what to expect when we put them in an aluminium box with an electric light bulb. But anyway, we went ahead and actually we tried both approaches. We did a translocation in July 2018 of 30 adults, which we released at our release site 1 in West Norfolk. And then 18 further adults, I took them back to where I live in Derbyshire and set them up in locust cages. And to cut a long story short, with a little bit of trial and error and tweaking the system, I came up with a system that really suited the large marsh grasshopper. So even though it is in nature, the adults are only found in very wet places, they do actually survive well in a fairly hot and dry locust cage, provided they're given fresh grass every day. That's absolutely critical. And sometimes they get fresh grass twice a day. And so they lay their eggs amongst the stems. The grass is provided in jam jars of water with cotton wool around the top to secure the stems in place. We use cocksfoot, Dactylus glomerata, which is a stiff grass which will stand up under the hot light bulb, and the grasshoppers can actually feed on it, and they can sit on it and bask fairly close to the light bulb if they want to. So it's got two good features there. And they lay their eggs amongst the grass stems, which I collect every second day, and then they can be processed and put into moist sand, and eventually they end up in the fridge for winter in egg diapoles, ready for use in the following year. So the system worked, and the 18 grasshoppers that I had, I got another 15 on a second short trip to the New Forest. They actually produced for me 265 egg pods by the end of September, which amounts to 3,500 individual eggs. So that was a great success, and we knew then that we could go on to the next stage in our plan, which was to recruit volunteers who might be able to help us in the next year in hatching these eggs and rearing them, but doing so at home in their own locust cage, and thereby spreading a lot of work between a lot of individuals to produce adult grasshoppers that can be released in the wild. So we call our volunteers citizen keepers, and we have to be quite choosy about who we take. We sometimes get a lot of people applying who are not really set up to do it properly. It's a job where you need to live somewhere with easy access to plentiful grass, especially when the grasshoppers are getting towards the end of their nymphal stages and into the adult stage. They do eat a lot of grass, so they need to have a ready supply of grass nearby. We prefer to have local people as our keepers, because obviously they've got a greater investment in the project. They're going to help to rear them and release them onto their own home patch, as it were. And when they've left the project, they're still around. They're still ambassadors for the project, and they can still get involved in other aspects of the project. So those are important factors. And also, we do have to be very vigilant about biosecurity issues, and so there's certain criteria that our volunteers have to meet. So we don't allow any of our citizen keepers to use flea treatment on their pet cats and dogs from 2 months before the start of the rearing season. We did have one disaster in the very first year where one of our keepers' parents had used flea treatment without the keeper knowing, and that did actually have a very detrimental effect on the first cohort of grasshoppers that she tried to rear. We don't allow any keepers who keep reptiles as pets, because reptiles often are fed on locusts and crickets from the pet shop, and there's a high risk of there's a risk of contamination of the grasshoppers with pathogens that can be carried by the locusts and crickets. And we don't actually have anyone who is keeping any other insect livestock in the house, again, to avoid cross-contamination risk. So the guys at Citizen Zoo, particularly Lucas Russo and Elliot Newton, did a fantastic job in our first year and coordinated the recruitment of, I think we had about 10 or 11 volunteers in the first year. And in June, I ran a crash course in grasshopper husbandry to train up the volunteers, and we do this every year now. And we basically train them how to feed the grasshoppers without them all escaping from the cage, how to select the right grass, how to set it up, how to set up the egg pods in the initial phase so that they'll hatch, how to use a pooter, which you can see about to be used here in this picture on the right. And at the end of the session, we give out the locust cages and other equipment that they might need, and egg pods. We also have a number of resources available for our volunteers. We have a training manual, which is now just provided online. It's downloadable from online, which covers everything in the training course and some additional information as well. And one of the things that works very well, we have a Facebook private group page, which allows any of the volunteers who have a problem or anything they're not sure about to raise it, and I check it every day and give expert input on it and explain what needs to be done. But also the citizen keepers can share information with each other, and we've got a really nice community of people helping each other through the Facebook page. And there is also banter, of course, which is good fun, and lots of sharing of photos. So when the training session is finished, the volunteers rush home to set up their cages and their egg pods. We use a sort of a filter wick system here. So the egg pods are in wet sand, but there's a filter paper wick feeding water from underneath so it doesn't dry out. And I've pre-conditioned the egg pods so that they should hatch after 3 to 4 days in a synchronized hatch, so which is important for us. So the eggs are sitting there, nothing's happening. There is tension, people starting to get worried, and then at some stage on usually the 4th day, but it could be the 3rd day or the 5th day, we get this happening. Is it going to do it? Yes. So here you can see a little video of some baby grasshoppers which have just hatched, and they crawl out like a little maggot from the egg pod. They've got a hatching membrane around them. They crawl up to the surface through the sand, and then the hatching skin splits and this tiny grasshopper climbs out. Good. So they climb up onto the grass nearby the egg pods and bask under the lamp or near to the lamp if they can get there. And it takes about 5 hours before they're all hardened up. And then they start it. By the way, at this stage, the Facebook page has gone into meltdown. Everybody is getting very excited and delighted at what's happening in front of their eyes. After about 5 hours, they start to feed. And for the next 3 to 4 weeks, the grasshopper nymphs do very little apart from feeding. There are 5 instars, and it takes about 3 to 4 days for each instar. So after 3 weeks, that we're getting to the stage where they're ready to do the final molt. This picture on the right here shows the 3rd and 4th instar nymphs. If you look at the picture on the left, you can see even at the small stage, they're capable of eating cock's foot grass, which is actually quite a hefty blade of grass, but they've got the equipment to deal with it, even in the first instar. And then we get to the final instar, and this is quite a spectacle. The grasshopper hangs downwards and pulls out of its 5th instar nymphal skin, aided by gravity here on the left. And once it's climbed out of there, it then climbs up onto a grass stem. And this is very much like a dragonfly pumping up its wings. It's sitting there hanging, hardening off and pushing hemolymph through the veins in the wing buds. And it takes about half an hour to an hour to pump up the wings. And again, there's a frenzy of activity and joy on the Facebook group page. It's quite a spectacle. So the first adult there, it's got fully formed wings. They 2 or 3 days later, the males will start stridulating. They start kicking out their hind legs and striking the end of the wingtips. And 2 days or so after that, we'll see the first matings in captivity. The molt to adulthood is usually finished after about 6 to 7 days. And at this stage, we usually get our keepers to mark the individual grasshoppers using a harmless colour-coded spot of oil paint. And this allows us to monitor the survival and dispersal of the grasshoppers after we've released them. So we do 2 releases a year, 2 cohorts of grasshoppers, first release in mid-July, second release in mid-August. So these are covering the whole of the natural grasshopper season in the wild. There's a sort of an early cohort and a slightly late cohort. And when we do a release day, we usually meet up in the car park at the nature reserve and walk out together as a group towards the release site. And when we get to the release site, we group them into 3 or 4 groups and release at 3 or 4 sub-sites all in the same general area. Now, the following slides just show a few of our citizen keepers, and you'll be able to see they're quite a diverse group of people. And they've got the grasshoppers in these net cages, which are our release cages. And it can actually be quite an emotional moment as well. It's a big thing, really, to let these babies go that you've reared at home for the last 4 weeks. They're very endearing insects. This was our very hottest release day ever. This is in July 2022 when we had the drought and the heat wave. We almost had to cancel the release because of health and safety concerns, but in the end we were able to go ahead and do it. But also, I should say, we have also released in the rain as well. It's not all sunshine. Now this slide just gives you an idea of the number of adult grasshoppers that we've released over the lifetime of the project. And I hope you'll agree they're pretty impressive numbers. The first year we just directly translocated the 30 adults, and then the next year was the first run at the home rearing, and we've learned as we've gone along. Really, we've fine-tuned the system, and 2022 was the drought year. We had trouble getting grass a little bit there, so I think the numbers were down for that reason. And 2023, 2024, both record years, consecutive record years. We're releasing plenty of grasshoppers. We've released at 5 sites where we've released adults. This is site 1, which was the initial site where we did the translocation. As you can see, this is a typical acidic valley mire in West Norfolk. Site 2 is reasonably close by to the first one. It's got more of a three-dimensional structure to the vegetation here, a mixed valley mire. It's slightly more complicated here in that we've actually got 3 separate areas with slightly drier habitat separating the 3 from each other. One of these sites is well established now. The other 2 we're still introducing there. Sites 3 and 4, we've introduced fewer grasshoppers at these 2. They're probably not quite suitable, really. But site 3 is a eutrophic swamp margin, and site 4 is wet eutrophic pasture land. It's got potential, but if you get too much rain, then it floods, and if there's not enough rain, it dries out. And there's probably not quite enough peat in the soil there to really to be a really suitable site. And then this year, for the first time, we released in the Norfolk Broads. Now, it's been 85 years since large marsh grasshoppers were seen in the Norfolk Broads. There were plenty of them seen this year. And this is a tall herb fen at Wheat Fen Nature Reserve. And this is really exciting because it's a very different sort of habitat to the main 2 sites in West Norfolk. And the adults certainly survived very well here. Although we feed them on a pretty boring diet of cocksfoot when they're in captivity, when they get into the wild, they go out and they're very a generalist grazer, and they love sedges as well as grasses, and they will even eat rushes. And they really tucked into the sedge bed. The females were clearly gravid and full of eggs. We've never actually seen them laying, though. It's a very cryptic and secretive act, the act of oviposition. But we can be sure they did lay eggs, and we saw several pairs mating. So the big question on all of our minds now is whether the eggs will survive and hatch next summer, which is really exciting. Now, we've done our releases. We do at our main sites. We do monitor the population afterwards. We do a mock release recapture exercise at the end of August, start of July. This to do it, we need sunny weather and a temperature above 24 degrees, in which conditions the males will start singing, they'll start chirping or clicking, and that just makes the whole survey a lot easier because you can find them more easily. If they're not, if it's overcast or the temperature's below that, then we really can't do a compatible survey in the absence of stridulation. So on the first day, we get a group of volunteers together, and equally spaced. Here we're about 5 meters apart each volunteer. We walk across, transect across the area, and we catch every grasshopper we see. And myself and one other person are usually running around behind the line, marking all of the grasshoppers with a spot of uniquely coloured oil paint. And then we release the grasshoppers at the same place where they were captured as the surveyors move on. And so the first day we're capturing, we're marking, and we're releasing them. And we're taking GPS coordinates for every individual grasshopper that's been caught. On the second day, we cover the whole bog with a series of parallel transects until we've covered everything. On the second day, we repeat the same procedure, but this time we're not marking them, we're just recording whether the grasshoppers we see are marked or unmarked. And then with that information, we can put the data into the Lincoln Index and calculate a population estimate. This picture just shows the findings from the 2022 and 2023 surveys at Site 1. And you can see the grasshoppers are fairly evenly spread across the bog. There are actually two slightly separated populations. There's a diagonal ridge of longer grass running diagonally from the top left to the bottom right of the plot. And so the grasshoppers are fairly well spread across the main bog in the centre and to the left. And in the top right-hand corner, there's one release site. And then there are some experimental plots in the top right-hand corner where we didn't actually release the grasshoppers, but they found their own way there. And these sites are flayed every winter to reduce the formation of purple moorgrass tussocks and to allow more sunlight to get through to the ground vegetation, the sphagnum. And so this is the main bog on the left here, and picture B on the right shows one of the experimental plots. And we didn't introduce there because we didn't think it looked that promising, but actually what's happened is the grasshoppers have found it for themselves, and actually that's the site with the highest population density. It's nice and squidgy underfoot. There's plenty of cotton grass. There's plenty of sphagnum there. There is purple moorgrass as well, obviously, but for various reasons, which we're not totally sure about, the grasshoppers are really thriving in that particular patch. For Site 1, over the last 3 years, the population has gone up from, estimated population has gone up from 375 to 414. And then this year, it's at least 500 and it could be 600. I'm still working on getting a best estimate. There were so many grasshoppers, we couldn't actually do the exercise in 2 afternoons. And I had to do a follow-up third day exercise with a rather small group on the main bog to measure the number of grasshoppers there. And we had to use a slightly different methodology, but I'm going to come up with an estimate, and it'll certainly be way over 500 and could even be 600. So every year, and this is after we stopped introducing, every year the population has gone up at Site 1. We're also collecting a massive ecological data on the large marsh grasshopper, on the longevity of the grasshoppers after release, on the movements, and we're also seeing quite a few mating pairs. As I say, oviposition is a bit of a mystery. It's a very cryptic act. The females get right down into the base of the vegetation. They're very well camouflaged, and we haven't really seen one yet. This next year, I think we're going to make a special effort and have a series of days where we really go for it and try and find them ovipositing. But I'm also doing some oviposition studies in the lab as well. The future plans, we're going to carry on monitoring our 2 main sites and hopefully get something going in our West Norfolk, in our Norfolk Broads site. We're looking to do further introductions in the Norfolk Broads and hopefully into the Cambridgeshire Fens in the future. And as I said, lab studies on oviposition and egg hatching. This is just a quick shot of the Citizen Zoo team who've put in a tremendous amount of hard work into organising the project and getting it going in the first place. And finally, a salute to all the citizen keepers who've put a lot of hard work into this project. But they've also had a great time, and an awful lot of them come back for a second year or even a third year or even a fourth year. So they have a good time as well as putting our hard work in. Okay, thanks very much.
Thank you, Stuart. That was a fabulous introduction to the work that's been going on, and wonderful to see so many members of the community involved in that as well. As we're sort of starting to run a little over time, we've got plenty of time for Josh's talk, but can I just ask any speakers if you'd like to write some of your answers in the Q&A? That might help us, so we've got time to answer everybody's questions, because we might not be able to do that live at the end of the session. So now I shall hand over to Joshua Stiles, last but not least. Josh is director of his own consultancy, Stiles Ecology, and he's also the project coordinator for the Northwest Rare Plant Initiative, and he's going to be talking about his work to reintroduce peatland plant species across Northwest England. So over to you, Josh.
Thanks, Jane. I'll do my best to keep to time. I'll put a stopwatch on. Hello, yeah, my name's Josh, and I'm a botanist. And so it was in 2017 that I began a conservation programme in the region of Northwest England called the Northwest Rare Plant Initiative, which is what I'm going to be talking about today. Initially, in 2017, this is really a project that focused on the reintroduction and reinforcement of some very rare species in my region of Northwest England. And of those species, of course, were a number of peatland specialists. And from 2017, what I began to do was reintroduce some of those peatland specialists across parts of Northwest England, particularly in the Greater Manchester area. And initially, all of those reintroductions, or most of them rather, should I say, were immensely successful. From things like lesser bladderwort that were extinct there for over 150 years, from 0 plants, in a few years the estimated population is over 2.5 million plants. Whitebeak sedge, from covering 0 metres squared, now covers hundreds of metres squared within and outside of the Manchester Mosses SAC and on other sites as well. The list goes on. But from that early beginning of reintroduction of rare species, one thing began to make me really think. And when it comes to restoration sites and targeting suitable habitats for rare species, what you'll often find is that the area of suitable habitat for oblong-leaf sundew, for whitebeak sedge, for lesser bladderwort are very often few and far between, isolated by vast swathes of unsuitable habitat dominated by things like Molinia and cotton grass. And so really what I want to go on to talk about is my approach when it comes to reintroductions on peat bogs. So to start with though, ombrotrophic bogs, which I'm going to focus on, rain-fed bogs, blanket and raised bogs, of course, are amazing. And as fundamental as they are, of course, I'm sure we all know on this call, they are at serious risk. They are some of our most globally important and globally imperiled habitats. We've talked about losing over 80% or damaging over 80% of our blanket bog, but over 99% of our raised bogs have been damaged or destroyed. There is an obvious and urgent necessity to restore them. Here is a beautiful example of an umbrogenous bog, an umbrogenous blanket bog I saw earlier this year. Fantastic habitat with an understory of sphagnum lawn and hummock with pools in between. And above the sphagnum, you've got ericoids, your cross-leaved heath and your heather. In pools, you've got white-beaked sedge. You've got all kinds of other species in there as well. And these are just the plants, things like bog myrtle, bog asphodel, brown-leaved sundew, lots and lots of different things. Really, really beautiful habitat. And although we tend to think of raised and blanket bogs as a singular habitat, of course, within our raised and blanket bogs, there are many micro habitats. So in a raised blanket bog, we've got bog pools and hollows, we've got sphagnum lawn and sphagnum hummock. And if we think about these micro habitats, this micro topography over time, over the ages, this isn't something that stayed static as far as bogs have existed in Britain. In damp and cool periods, what you'll find is you have bits of sphagnum lawn, hummocks, and plenty of bog pools. And this is really important, I think. And of course, in drier and warmer periods, those pools that carry that excess water are much rarer in terms of a microhabitat on a bog. So drier, warmer periods, we have a lot more lawn-forming, hummock-forming sphagnum. And this brings me to restoration approach. When it comes to restoration of our blanket and raised bogs, what I've found, what I think, is that really current guidance, although there is this overwhelming focus on improving hydrological function, which is so important, from gully blocking to ditch blocking to bund creation to topographical adjustments, there is very often, 99 times out of 100, a massive understating of the importance of restoring not just sphagnum, but distinct assemblages of sphagnum on raised and blanket bogs. And I'll get more onto that in a second. Because when we look at re-wetting, as important and fundamental as it may be to the restoration of blanket and raised bogs, re-wetting alone does not solve all of our problems. When it comes to re-wetting alone, it doesn't resolve nutrient flushing. When we think about drying degraded bogs, what we'll often hear is they release tons and tons of carbon. But what we won't very often hear is they also release tons and tons of nutrients, especially extractable nitrogen. As well as the re-wetting's inability really to resolve those issues with nutrient flushing from the degraded peat, those nutrients that are seeping out. Re-wetting alone does not re-establish those specialists, those peatland specialists, with a poor, Jane was talking about reintroduction being important for those species with a poor ability to disperse. Re-wetting alone, if we re-wet a bog, once we've wiped out those species with a poor ability to disperse, are they going to come back of their own accord? The answer is either no or yes after several lifetimes. And so as a consequence, what we'll often see is that raised bogs and blanket bogs in restoration are often species poor and far removed from their undamaged and near natural counterparts. And when we look at the impacts of nutrient flushing as well, you'll see these monocultures of purple moorgrass and sometimes common cotton grass form as a result of that nutrient flushing from the degraded peat. And although we might be planting some sphagnum species, especially things like Sphagnum cuspidatum, Sphagnum phallax. These are species that often recolonize, they can recolonize quickly, sometimes naturally. Although re-wetting can encourage the colonization, the recolonization of those hollow species, these are species that are not strongly associated with the ecosystem functions that we desperately need, be it nutrient sequestration to tackle things like the formation of millennia, monocultures. They're not strongly associated with optimum hydrological function, and nor are they associated with the formation of those important features on a bog in drier and warmer climates, lawn and hummock. These are microhabitats on a bog that give them resilience in drier and warmer climates. Why does it matter? Sphagnum are surely all the same. Well, no, they are not. Of our about 35 odd sphagnum species, only 5 are associated with deep peat formation. And the reason for that is because these are species that produce a heck of a lot of phenolic and other acids, and they're also chunky generally, or they're very compact in their growth form, which means that they're great at water retention. And both of those adaptations give these 5 species incredible resistance to decay, and therefore they're associated with substantial peat formation, which other species are not. However, of those 5 species, only 2 are typically abundant on healthy raised and active bogs. They form a substantial component of acrothelm or peat forming vegetation, and that especially includes papillose bog moss, which can form up to 90% cover on healthy, active, raised and blanket bogs. And that really matters to consider these 2 species, because ultimately, if we do not have peat-forming species, especially papillose bog moss and Magellanic bog moss, we are not going to address key problems that our bogs in restoration face at the moment. Both of these species are associated with nutrient sequestration. We think about bogs in the context of carbon sequestration so often, but we seldom think of them as being important for nutrient sequestration. Nutrient flushing is a huge problem on a lot of our raised and blanket bogs. And so these are species that are able to form peat, sequester nutrients to the detriment of negative indicator species that are testament to a degraded habitat like Millennia, purple moorgrass. They're chunky, they're able to retain water in drought periods, which our hollow species can't. As soon as a bog pool dries up or a wet area dries up in summer, they desiccate so easily. Our Sphagnum cuspidatum and Phalax, these don't. Papillose bog moss and Magellanic bog moss don't. And as a consequence, as they reform sphagnum lawn and hummock, they're associated with much improved hydrological function, unlike other species which more readily colonise sites. And of course, being able to keep sites wetter during drought periods, these are species that are ultimately responsible for instilling climate resilience compared to all other species of sphagnum. So just talking about some of the things I've been involved in the past few years, as well as those rare plant reintroductions. Papillose bog moss on the Manchester mosses, this top right picture is one of them, what one of them looked like. Most of our Manchester mosses are former peat extraction sites. And if we think about how these sites would have looked in the past, papillose bog moss would have dominated the surface of a lot of these habitats, which ultimately creates habitats for a huge number of plants and animals. And so over the past few years, this is a species alongside those rare plants that I've been involved in the reintroduction of over in excess of 8 hectares across Greater Manchester. It is an absolute keystone species. When people start talking about beavers, just put your finger on your lip and say you haven't heard about Papillon's bog moss. Marvellous. Anyway, so here's an example of one of the little plugs that I've been monitoring over just a few years. So we've talked about sphagnum growth already today, so I won't go too much into it. But year one, got 18.8 centimetres in a year. It's grown another 10 centimetres. And we can see here in year 3 these plant sphagnum papillosum plugs that have been planted close together. They are expanding and they will coalesce, hopefully, and form sphagnum lawn once again in the near future. Alongside translocating sphagnum papillosum, which I did from active, healthy sites nearby, putting it onto a degraded site in a responsible way with all the permissions, might I add. Another thing I've been involved in is actually mapping this species across the Manchester mosses so that managers know where this plant is. And that's really important, because not only then do land managers know where we've reintroduced this plant to, where we've re-established this plant, but we know where large hummock and lawn exists already, so that we can use those points, those lawns, those hummocks, harvest from them and disperse that species further afield on these sites. One thing I will add is we've translocated this keystone species over in excess of 8 hectares in Greater Manchester. But one thing about transplanting this individual sphagnum species is when we are transplanting this species from an active site, from a healthy site, it isn't sterile. It isn't just that species that we're transplanting. We could be transplanting in a handful of sphagnum papillosum 5 different leafy liverworts, lignophiid spiders, all kinds of other invertebrates that are associated with sphagnum carpet. Other thing that we definitely have transplanted alongside these sphagnum plugs are lots of specialist species, many of which were formerly extinct on the Manchester mosses SAC. From bog rosemary, a beautiful little ericoid, to golden bog moss, wild cranberry, bog asphodel, white beak sedge. Should be the most important, most visible plant of bog pools on these healthy raised bogs, went extinct. Bog myrtle, often known for it being a mosquito repellent, but what people don't often know is it contains abortifacient, so it can induce abortion if you have enough of it. Be careful. And things like round leaf sundew. So it's not just these sphagnum plugs that we're putting back. We're putting back key vascular and other plants that form important components of healthy, active, raised bog. So I think I've stuck to time, hopefully. To conclude, sorry if I've whizzed through that. To conclude, reintroductions of species, as Jane said at the start of this presentation, with a limited dispersal ability can be, and often is, an essential part of peatland restoration. These species aren't going to restore, recolonize themselves a lot of the time if they've been lost. Reformation, in particular, of peat-forming sphagnum lawn, not just any old species, Papillose bog moss and Magellanic bog moss, is an essential action to restore active peat-forming vegetation that looks anything like the habitat we've lost. But not only that, it's likely to be very essential for our bogs to survive in a drier climate. One thing about sphagnum transplants as well, of course, is unlike planting a tree and having to wait, I don't know, a couple of decades to yield to get a bucket full of acorns, sphagnum grows laterally and it can grow very fast. So I think really when it comes to establishing sphagnum, very quickly you can transplant something and that little clump, that little lawn, that little hummock can become a donor site in and of itself. So we can use it to plant even more in the future. I hope that's been interesting. And I think that is everything. I'm not sure. I'll have a look if there's any questions. Oh gosh, I'm not sure if there is. Anyway, there you go. Thanks.
Fabulous. Thank you so much, Josh. That was a wonderful whistle-stop tour. But yeah, really important to think about, you know, it's not just about getting these species back. It's about restoring all of that structural integrity and everything that that means in peatland restoration. So we do still have a few minutes left. So I think there's a question that has come in for Josh now. So if anyone's got any more questions for any of our presenters, most of them have now answered them in the Q&A. So thank you to our presenters who've done that. But there's a question for Josh. Do you consider the role of reducing nitrogen and methane emissions when you reintroduce certain species, or is the main priority for carbon sequestration and ecosystem function?
Not so much methane, but when it comes to nitrogen, nutrient sequestration, absolutely. So as I said sort of at the beginning, my initial approach to reintroduction of those rare species was to target these pockets of habitat that are already suitable for some of those rare plants. But when it comes to Papillo's bog moss reintroduction, this is always in habitats really that are unsuitable generally for those rarer plants, with the aim of ultimately making them suitable by nutrient sequestration through instilling better hydrological resilience in areas that, I don't know, might be mostly bare peat with a bit of common cotton grass or heather. So I mean, yeah, restoration of ecosystem function is great, but yeah, nitrogen nutrient sequestration is yeah, really important consideration. It's kind of what instigated the urgency, for, in my mind anyway, for all of these reintroductions that we've coordinated over the years of Papillo's bog moss. So yeah.
Fabulous. Thank you, Josh. There is another question that's come through. So this might be one for Tom or any of our other speakers, because I think, you know, moorland, very broad term. How do you make the average person excited about moorland protection? And public engagement feels like it will be key for the future. So I think we've probably all got something to say on this, but I'll hand over to our speakers. Anyone wants to step in and answer that one?
I can say a couple of words on that. So I think it's a really good point. And I think, as with most things to do with peatland restoration, we need to be doing way more than we are. So I think that's in terms of the capital works and the research and the communications and engagements. We need to be like massively stepping up what we're doing onto a hugely bigger scale. I think, you know, at Moors for the Future, we've had some pretty good success with communications events, you know, doing stalls and talks at events, and, you know, having stands at festivals and, you know, that kind of thing. My perception is that mainly the people who come up to a stall or a stand like that are people who already broadly agree with, you know, what we're talking about, and potentially already know a bit about it as well. So I think it's easy to reach people who we don't really need to reach. It's much harder to reach people who we really need to reach. So I think doing stuff in schools is good for that, you know, starting as young as possible. Yeah, trying to get sort of information out onto like the biggest platforms possible, you know, where we can reach the, you know, the biggest number of people. And I think it is just very difficult, you know, like we do have relatively good engagement figures, but on a population scale, we're not talking to enough people, I think. But talking to people from a young age, getting people out to see sites, that's what gets people interested in and enthusiastic. But it's a really important problem for us to be looking at as a wider community, I think.
Yeah, and the peatland programme made real efforts this year during our annual conference to try and highlight some of those more creative approaches to engaging people as well, because, you know, we need to use all the tools that we've got in our arsenal to get people excited about this stuff. So some people might respond more to artistic approaches, some might respond more to really engaging ways of putting the science to them. So I think it's about being adaptive in our communications approaches too. So there's one more question, or a couple more questions. Let's see if we can get through the last couple very quickly. So a question for anybody and everybody is, for large-scale peatland restoration jobs, it seems that sphagnum plug planting input across large sites would be very labour-intensive. So what are your opinions on the most accessible species for reintroduction from a private sector approach to peatland restoration? So I suppose getting the most bang for your buck is what this question is about. Anyone like to step in and answer that one?
Yeah, can I?
Of course.
I think restoration of your sort of blanket and well any mire really, it's like chopping down an ancient woodland and bulldozing it completely and then trying to recreate it, is how I see it. I don't think there's any easy way of doing it, that's not costly or labor intensive. And actually, I think plug planting from commercially available stuff is probably the least desirable way to do it, because within micro-propagated stuff, as essential, as important and useful as it is, it's a sterile thing that we're getting, really. We're just getting those mosses. We're not getting the vast assemblage of other species that come in an active sort of raised or blanket bog. So that's how I see it personally. Yeah.
Can
Yeah,
I
there's
add
no easy
another?
outs.
Can I add another angle to that? So I think I totally agree. We need more than just sphagnum for sure. The scale of the problem is such, and the kind of the urgency of the climate crisis that we're in is such that we need to be tackling this problem at enormous pace, way faster than we are. And if one organisation like More to the Future is planting 2 million plugs a year, and we recognise that we need to be going faster than that, then I think translocating sphagnum from existing sources there's just not enough of it. Certainly in the peat district, there's just not enough of it. So I think planting plugs is kind of necessary. I totally agree that it's not ideal, but I think it is necessary. And our approach to which species to plant is like you say, it's restarting an ancient woodland from a bulldozed field. So we kind of recognise that there are some species that we really want to get there, like papilosum, like you've mentioned, the other ones that are the key peat formers. But we might not be able to go there straight away. Like that would be ideal. But so in our species mixes, we include papilosum and the other peat formers, but also some pioneers, which don't have such good function as you described. So things like phalax and palustre, but they're great pioneers. And from sort of paleo records, we've seen that often you get phalax, palustre, and then you get the other more sort of key peat forming species following on. So I think in terms of, and the advantage of planting sort of multiple species plugs is that within that plug, you'd hope that wherever that plug goes, as long as it's within a broadly reasonable window of conditions, that at least, one species will find its niche there. So there, you'll have the phalax that gets going first, but then also if it is suitable for papilosum will come in there. If it's more suitable for capillifolium, that one will come through. So, but I think by kind of going a broad sort of mix of species, you allow the opportunity for the right species for that condition to find its niche and get going. It's not an ideal way to do it, but I think at the scale that we're talking, we sort of have to do something that's practicable at scale and at pace.
Yeah, I think the other thing to mention is actually, yes, this work is expensive, but if you value that in terms of the ecosystem services we're getting and the benefits that brings, and how cheap a solution it is on our journey to net zero compared to other options, you actually do get an awful lot of bang for your buck with peatland restoration, if you like. And I think maybe there's a role for us as a community to make sure we're really communicating that well as well, and encouraging people from the private sector to understand that it is a really good value option. So I think we will have to wrap up the questions there. I'd like to end with a really big virtual round of applause for all of our speakers. I hope you'll agree that's been a fascinating range of talks today. I've certainly learned a lot. And I hope it's been useful for everyone in our audience too. So thank you to all our speakers for sharing your time and expertise with us today. It's really appreciated. So as I said, this webinar has been recorded, so we'll make it available on the Peatland Programme's YouTube channel shortly, where you can find our previous biodiversity webinar. And if you'd enjoyed this one, you might be interested in a series of webinars that we're just starting to pull together for next year, in collaboration with the Environment Agency and with a water theme. So I hope that's piqued your interest. Keep an eye on the events pages of our website, because we'll be starting to announce those and calls for presenters soon as well. So if anyone has an interest in those hydrological aspects that we've covered today, then keep your eyes peeled. So I will let everybody go and have their lunch. Thank you again to all of our speakers, and please do stay in touch with us as well. Thanks, everybody.